All right. How is a registration going?
We're registered.
Good, so. I just want to assure you, if you want to take 102, you will get into 102, even if there's not a spot currently available. All right. If there's not a spot currently available, register for the limbo sections, and then we will make sure to add seats to a section that works in your schedule. All right. The caveat to that is if you are in sports, we will do our best to get you in a section that does not conflict with your sports practices, but we cannot guarantee it for sports. Right? But long story short, if there's nothing open, register for those limbo sections. We will get you in. All right. Any other questions that I can answer potentially about registration concerns? All right. Sounds good. I know registration as a first year can be particularly troublesome, but I promise it gets easier the longer you are in your academic career. All right, so we're going to start with our last bit of information that will be covered on the exam today. And it's really just a continuation of Mendelian genetics that we've already talked about. All right. And so we're really going to start off by talking about a two-factor cross. So we did a one-factor cross in lecture on, gosh, is it Wednesday? It is Wednesday. On Monday. I keep thinking it's Friday, and I don't know why. But today we're going to start with a two-factor cross. So two-factor cross, we are following two different traits at the same time. All right. And so Mendel did this for his pea plants. He would choose two traits, made sure he had true breeding pairs for those two traits, and then did a two-factor cross and followed what the results were in the offspring. Right? Now, if we do a two-factor cross, there can be two possible patterns. Right? One pattern is that these genes could be linked. All right? So if we find these genes together, right, you're always going to find them together. Right? So they're always going to be transferred from the parent to the offspring as a unit. Right? So they would be linked together. We'll talk about that a little bit further. But the other possibility is that these genes are independent, which so far has been most of what we have seen in lecture in lab. Right? They're independent. They're going to be randomly distributed into genes. Right? They don't have to be inherited as a unit. Right? So often, if we're looking at chromosomes, right, if traits are linked, they are going to be on the same chromosome, and they tend to be really close together. Right? So this chromosome on the bottom shows some genes that are linked. They're on the same chromosome. They are close together. This one on the top, right, these two genes are not linked because they're on different chromosomes. Right? So this physical association of at least two genes, it can be more than two genes, but at least two genes on the same chromosome is called linkage. All right? And so linked genes, they're always going to be transmitted together as a unit. Right? We're never going to transmit one gene and not the other. Right? Both genes will always be transmitted together. Okay? And it's going to violate that principle of independent assortment that we have been talking about. Right? So they are no longer independently assorted. And again, a linked gene is going to be really close together on the same chromosome. They're on different chromosomes. They will not be linked. They can be on the same chromosome but far apart and not be linked. But if they're close together, same chromosome, that is a good indication of linkage. All right, so let's walk through some examples of what we might see if the gene is linked. All right, so if we cross two true breeding pairs, all right, so the first one is true breeding for the dominant genes. Second one is true breeding for those recessive genes. Right? The only possible combination of alleles that they can pass down, right, is either going to be big Y, big R, or little y, little r. And they're going to create this F1 generation that is heterozygous for those genes. We have seen this thus far, all right? So this is nothing new, right? But after this F1 generation, this is where we can start to pick apart linkage. So this F1 generation, right, is only going to be able to pass down those alleles that are linked together as a unit. So because big Y and big R are linked, this F1 generation, even though it has the recessive alleles, can only pass them down as these linked big R, big Y. Right? That's the only combination that can be passed down because they are linked. Right? Same with the little letters here, little y, little r. Right? The only way that this F1 generation can pass it down is as a little y, little r, because they're linked together. Right? And if you think about this in terms of being on these homologous pairs here, you see that R and Y, they're close together on the same chromosome. Right? And this F1 generation, they have all of the alleles because they have a homologous pair, right? But those alleles are going to be linked together on a pair. So we're not going to transfer just one capital letter. It has to be both of them at the same time. And so that F2 generation, we're only going to get a few of those combinations that are possible, right? Which is different than what we've seen in our two-factor crosses, right, with independent assortment. So in independent assortment, right, these letters don't have to stay together because they would not be linked, and therefore they can be split up into different gametes, right, and we can have many more possibilities for this F2 generation. Right, so in this second example here, it's likely that these traits are either on different chromosomes or they're far apart on the same chromosome. Right? So if we look at the outcomes of our two-factor crosses, like I said, there could be two possible outcomes. It can be linked or it can be independently assorted. Right? If we look at the outcome of a two-factor cross that Mendel did, right, we can see that when he crossed these yellow round seeds with green wrinkled seeds, right, he got yellow round seeds for the first generation, and that second generation, right, all the different phenotypic combinations here, right, ended up to be in a 9:3 to 3:1 ratio. Right? Now Mendel saw this for all of his plants because he just so happened to be looking at all traits that were not linked. Right? So Mendel would see this 9:3 to 3:1 ratio, and that led him to come up with this principle of independent assortment. Right? So if genes are independently assorted, we will always see this ratio appear. Right? This is not always going to be the case, right? But in this case, what Mendel is doing, it was. All right, so I just want to walk through how to set up that two-factor cross in a Punnett square. I know you did this in lab, but this is just a quick refresher here. So if we're crossing two individuals of that F1 generation, we first have to determine all of the possible allele types. All right, I just realized I don't have closed captionings on. I also forgot that on Monday too, and nobody said anything, so they are now on. All right, so we have to figure out all those allele combinations that could be passed down in the gametes, right? And so basically we have to go through and figure out all those possible combinations, all right? And so if we are only looking at these two genes, right, there are going to be 4 possible allele combinations in the gametes. Right? You'll then put these combinations for one parent along the top and for another parent along the side. Right? Match all of our letters together, and because this one's independently assorted, we would find that we have a 9:3 to 3:1 ratio for their phenotypes. All right. So Mendel always saw this 9:3 to 3:1 ratio, right? But that's not how all traits are going to end up looking like after a two-factor cross, right? Because some traits are linked. And so if they're linked, we're going to see something other than that 9:3 to 3:1 ratio. And a really good example of this is Bateson and Punnett So yes, the same Punnett that developed the Punnett squares, right? They actually looked at sweet pea plants, right? And they looked at the pollen shape, either long or this round pollen, or purple versus red flowers here. They did the same type of process, right? They used true breeding pairs. They got the F1 generation. They bred this generation together, right? And they got this F2 generation that followed this ratio of 15.6 to 1 to 1.4 to 4.5, right? That is not a 9 to 3 to 3 to 1 ratio. And so what they found, right, is that these genes are actually linked, right? The ratio that they observed is very different from what they expected, right? And if we wanted to confirm this, right, we could do our chi-squared analysis like we did in lab, and we would see that we would reject our null hypothesis that there is no difference between our observed and our expected, right, which tells us that these genes are linked. So we're rejecting that hypothesis of independent assortment. All right, let's do a group question here. I want you to look at these two examples and figure out which one is independently assorted and which one is linked. All right, pay attention to those ratios at the bottom. Yes, I had to use a cat example. You good? The cat is very frazzled. Oh, and it doesn't show up very well, but this cat here actually has orange hairs sticking up. Yeah. Yeah. It didn't want to color the whole thing orange, but it had the orange hairs, but in the projector, you're not seeing that. This should be a fairly straightforward question here. I'm pretty sure this orange cat was an AI-generated image. If I don't know if you've seen like those Pixar challenges going around, I feel like this is part of those Pixar challenges. Looks very cute, right? Like Pixar-like. All right, which one of our examples here would be independently assorted? Yeah, the first one, the green one here, right? We see it follows this 9 to 3 to 3 to 1 example, right? And the way I set it up, it follows it pretty perfectly in the 9 to 3 to 3 to 1. We can have slight deviance from that, right? But we have our 9 to 3 to 3 to 1 ratio, which means this other one, which has a, I believe I set it up as a 1 to 7 to 7 ratio, 1 to 7 to 7 to 1 ratio. It's not 9 to 3 to 3 to 1, so it is linked, all right? Are there any questions about linkage? All right, we are going to expand upon that topic because a lot of times linkage is found in our sex chromosomes, right? So sex chromosomes are found in many species, but not all species have sex chromosomes. So a little fun fact, so like species that can be hermaphroditic, like snails and earthworms, they don't actually have sex chromosomes, which are interesting. You have a question? They could be either male or female or a combination of both, or they can change between it. Yeah. So most species have sex chromosomes. Right? There are several mechanisms for sex chromosomes. We're familiar with the XY system because that's what we as humans have. All right, so males are XY, females are XX. Right? Insects use this X0 system. Right? So insects could either be XX or just a singular X. There's actually no Y chromosome there. Right? Other individuals, particularly like birds, will use this Z to W system. So males are ZZ, females are ZW, right? just different ways of writing out the sex determinations of different organisms. There are many more systems than just these three. Just thought I'd point them out because it's interesting, right? We always think of sex chromosomes as X or Y. Not always going to be the case in the animal kingdom. So in humans, though, right, we are going to stick with that XY system. The X chromosome tends to be larger, and because it's larger, it can carry more genes, which is often why a lot of genes that are sex-linked are linked to the X chromosome. Purely because it's larger, it can carry more genes. Right? So we would say males are hemizygous for the X-linked gene because they only have one X, right? They also have a Y, right? Females have two X's and 0 Ys. Right? A really good example of sex-linked traits is hemophilia. And I know you worked through this in lab, but it's such a great example here. So hemophilia is a blood clotting disorder, right? It is a recessive disease, which means you have to have the recessive traits being expressed, right? And so if we were to look at some chromosomes, right? So this female here is a carrier, right? She has a dominant allele and a recessive allele. So she carries the allele for this gene, but because this allele is masked by the dominant allele, it's not expressed, so she doesn't have the disease, right? Versus this male here, right, has the recessive allele. It is not masked by a dominant allele, so it is therefore expressed. So this male has hemophilia, right? So there's that distinction between being a carrier and actually having that disease, right? This disease is actually prominent in some royal family lineages, as we see up here. So Queen Victoria was actually a carrier for the disease. We know she's a carrier because it's kind of split in the gray and white in that circle there. So she has it, she did not express it, right? So she would be heterozygous for that allele, right? Her husband did not have it, Albert, right? Their children, she was able to pass down that allele to her daughter, who in turn passed it down to her daughter. And finally, by the time we get to the mating of this Tsar of Russia and Alexandra, they have a son, right, that won the luck of the draw here and actually has expressed hemophilia, right? Yeah. So females could have hemophilia like this one, or any X-linked disease, but they would have to have little h and little h if it's a recessive disease. Yes, yes, yep. So their father would have to have it, which is why, I mean, good example, if we look in this family tree here, none of the fathers had hemophilia, so none of the daughters were able to have hemophilia, because you'd have to have two recessive alleles, and in this combination, you only have one. But males could have it because males only have one X chromosome, so it's easier to be expressed in males. Right? So when we're thinking about these X-linked diseases, right, we could set up a Punnett square in the same exact way that we've done before, but we just wanted to note which chromosome is the X and which is the Y. So if we were to cross two individuals here, right, so our female with two X's, our male with an X and a Y, we would set it up the same way. We would just also have to keep track of the X and the Y. And because the Y does not carry that gene, it's never going to have an allele on it, right? If it's an X-linked allele. Right? Making sense so far? Any questions? Yeah.
I was gonna ask if
Yeah. I mean, it would kind of be the opposite, right? But only one allele would be on the Y, and that would be the only allele that would have or be present, right? Because there's only one Y in the cross. Yeah. But they're much less common just because that Y chromosome is so much smaller. What was that? I would be surprised if they don't. I don't have an example off the top of my head. But just like all DNA, we could have some variations in our alleles. I would assume there are Y-linked diseases. All right, so there are other types of dominance out there. All right, so moving away from the X-linked dominance, right? It's not always as black and white as one being dominant and one being recessive. Depending on the gene we're looking at, we could get different phenotypes showing up. So one of these is incomplete dominance, right? So when we have this paternal generation, so we have a white flower and a red flower, when they mate together, they produce this offspring that actually has this blended phenotype, right? So not one allele is being expressed or hidden, but rather they're kind of blending together to create this pink color, right? But if we were to breed these and get an F2 generation, we would see the white and the red reappear over time, right? So, good example of it not always being just dominant or just recessive.
Does that mean that now on its own, it's
So pink is a phenotype.
But does it become one like it's white and red? Does it become like one of those that is possible every generation?
So in this instance, right, if we have a R and a W together, right, those two alleles blend together to create this pink. But this R and this W, right, if we were to take this F1 generation and breed with itself, we could once again get a combination of just W's or just R's, which would lead to the red or the white showing back up. Does that make sense? So we're not changing what alleles are present, right? It's just the way that they're expressed are a little bit different than fusion here. We can also have, there we go, codominance, which means we don't have any that are recessive. We don't have those that will blend together, but rather both of the alleles are going to fight to be expressed. And we might see something showing up that's a little bit of each, right? In this cow example here. So they're not blended to create a new like color phenotype here, but rather both phenotypes are being expressed in some way on that individual, right? So in this example, it's the spotted cow. We see the brown, we see the white fur coloring, they're together. It's not like it's a light brown overall color, right? That would be incomplete dominance if that showed up. All right, we could also have pleiotropy, right? Or pleiotropy, I think pleiotropy is how it's pronounced. And that we have a gene, right? But that one gene can influence multiple traits. So it's not a gene, a trait, it's a gene, multiple traits that it's influencing, right? So Marfan syndrome is a good example of this. So Abraham Lincoln had this syndrome where he had one gene influence how tall he was. He had disproportionately long limbs and fingers. He had an abnormally shaped chest and some heart problems. All of these traits showed up from this one gene. Right? So again, not as simple as black and white, dominant, recessive. Right? We could also have traits that are polygenic, meaning that there are multiple genes controlling for a phenotype. Right? So in corn, for example, this variation where we get this red corn, right? This coloring is the combination of 3 different genes and how they blend together. All right, so instead of a 2-factor cross, this is essentially like a 3-factor cross where every single different type of genotype shows up as a different phenotype, which is why we get this like these different hues of red showing up depending on the genotype. Right? It's these 3 genes all working together to create one phenotype. So kind of the opposite of pleiotropy, where it's one gene, multiple traits. This is multiple traits, multiple genes. Right? All right, so we can kind of describe these different types of traits as either being qualitative or quantitative. Qualitative is what we are most familiar with, right? So they're very discrete phenotypes, right? You have yellow or purple flowers, right? Or you have, you can roll your tongue or you can't, right? Very distinct phenotypes. The ones that we just kind of went over in brief detail here, where we can have an influence of phenotypes, right? A distribution of phenotypes would be quantitative, right? So that would be some good examples are height, skin color, weight, but also that red corn example that we have, right? That would be a good example of these quantitative traits, which people tend to be less familiar with because they're more complicated. They're harder to understand. Yeah. Why do they have these desires instead of one or the other? Something to do with its genetic code? I don't know why a gene would show up like that, or why it might be beneficial, or why it evolved to be that way. I don't know. I'd have to do some research. All right, my last slide here, right, is that phenotype. We have talked about it being influenced by allele, but we can have environmentally influenced phenotypes as well, right? And a really good example of this is pigmentation of rabbits, depending on what temperature that they were reared in, right? So rabbits here, right? We have our rabbit up top here. If they are reared in this warm temperature, so above 30 degrees Celsius, right, that actually inactivates the pigment gene, so that gene product is not being made, and it leads to rabbits that have this non-pigmented white fur. Right? The opposite could be true for those reared in these colder temperatures, so less than 20 degrees. Right? If they're reared in these colder temperatures, that gene is activated, the gene product is made, and we get some pigmentation on the fur. Right? Another example of this, I believe, is sea turtles, right? Depending on the temperature of the beach that their eggs were laid in, right? That temperature can determine whether they are males or females, right? So there's some environmental control over some phenotypes, which is very interesting. There's other examples out there, I am positive of it. So if we were to look at this, which part would be gene regulation, or which part would be gene expression? So what part of this diagram am I regulating my genes? Yeah, we're regulating whether they're being turned on or off. Yes, so gene regulation. I probably gave you these in your slides. Yeah. Which means gene expression is whether or not that gene is actually expressed or the product is being made, right? That protein is being translated. All right. Any questions? Really wanted this lecture to be short because I didn't want to give you too much information before the exam. So your scientific literacy assignment will be due on Friday. Friday will be mostly a review session in class, right? I do have a couple of extra, I think it's one extra sheet on transcription and translation if you want to practice that, right? I also have a practice review question sheets that will become available on Friday during class time that you can work on during class. All right, the review session, I'm going to hold an extra one, and this room is available at 3 o'clock on Friday, so I will be here from 3 to 4 on Friday if anyone wants to attend that extra review session. All right, our content check. All right, I'm going to have you make a 2-factor cross. All right, so I want you to draw the Punnett square for this 2-factor cross, and I want you to also report what the genotype ratio is, or the genotypic ratio is, and the phenotypic ratio. Right? And I promise it will not be as complicated as the one in lab. And somehow my closed captioning is just... So, anytime an assortment is probably randomly aligned. And so how they randomly aligned is going to screen that and use the So I will say that these are not linked. But they're limited in what combinations they can pass down because they just don't have difference in over it. That's it. You could like draw an underline underneath like this. You can have a capital and a lowercase after one underneath. You could have, once again, two capitals. And once again, a capital and a lowercase. These are not linked. We're still going through meiosis. We're missing 4 gametes. Remember, in that two-factor cross, we're crossing 2 letters out. So on each row of the column, we should have both of the rows. So, I think I already set it up. I want to make sure I have a T and a P for every column and every row. Same way I got those. I figured out all the possible combinations for the 4 gametes. So I put that on one side, so that's one gamete. Okay. I mean, you could say, okay. Yeah, so you use a Google, what'll happen is someone will contact you and figure out how to do it in other. You will make it at work, but you might have to be flexible in your schedule and what you want. Yeah, yeah, yeah. If you don't take that least, if you were to say what the ratio is possible, then you would have to say it's 0. So I would say that because it's what we are seeing. I want you to get that the G is on both of those. One is on one is on the other. Or you say G, but you're in a different period. Yeah. Yeah, and it's basically a brace right? So if you simplify this down, half to half, it's just 1. Yes. Yes, yeah. For a couple flowers, it could be so go to figure out what else phenotypes. How many of those how many of yeah,
Yeah, I know. That's not a meditation. I'm not a sensitive. I don't have that gene. Just kidding. You know, there is a gene. There's a taste bud that actually harness metabolizing. But it's just like something.
yep. It is an equal amount, yeah. Okay, and if you put that into ratio, it would be. So that's for which ratio? That's what we see in our cross. If we were to say of all the different possible phenotypes, okay? You could also say it's like a 0 to 8. 0 if you pick the one.
I like this part. It's good. I do